I didn't make for you a place to upload the data. I actually meant to. I think I did in a later version and then didn't keep the changes, or I don't know what happened when I was uploading, but I don't think I uploaded the changes that I meant to. So the first thing you need to do is load in your data. There's a couple ways you can insert a code chunk, or you could have just done this here. It doesn't really matter. I like to press Control or Command, Alt, and then I, and it inserts a code chunk. We use the file.choose function to select our files. I'm selecting the Lab9 warm-up CSV, getting the output for that, copying the whole thing. And then I'm just making a warm-up object using the read.csv function and pasting the file location that I got, and then using the head function to check what the warm-up looks like and make sure that it uploaded correctly. So yeah, we've got shaded and sun exposed, which matches with the word problem that we have. So we're comparing the abundance of insects or beetles between a shaded and a sun exposed pond. So let's see, it looks like we have some a priori hypotheses. We believe that in the sun exposed location, there's a less hospitable environment, so we expect to see fewer beetles there. So you might find that it's actually easier to write your alternative hypothesis first, since it's the one stated in the word problem. So I'm going to do it that way. So X bar sun exposed is less than X bar shaded. So essentially we expect there to be more beetles in the shaded area compared to the sun exposed area. Which means our null hypothesis is everything else. It's the opposite and has that equal sign in it. So X bar sun exposed is greater than or equal to X bar shaded. This is an unpaired t-test, which means that we do need to test for equal variances. So var.test, remember that's the function we use, warm up. So I'm selecting the object, $, and then selecting the two columns. $, oops, not var test, warm up, $, sun exposed, and that's everything that goes in. So I'm pressing Control Enter to run it. And I get my F calc, which is greater than 1, which is good. That's what we want. We want the right side of the distribution. So our F is equal to 2.0089. We have degrees of freedom, our p-value. So we will accept the null because our p-value is greater than 0.05. Right? Our p-value is like 0.1. So that tells us that the test we're going to be doing is a homoscedastic one-tailed t-test. So doing the test, we use the t-test function. So warm up. Remember, when it's highlighted like that, I'm just using the tab key to kind of autofill it. It's just much faster. Our dot equal is the argument we need to use to tell R that our two treatments are equal to each other, or the variances are equal. It's true. And alternative equals greater, because we expect the shaded region to be greater than the sun exposed. Running that, we get our t-calc and our p-value. Statistically, looking at our p-value, it means we can reject the null hypothesis because our p is less than 0.05. Oops. Biologically, we could say shaded regions have higher beetle abundance compared to sun exposed ones. Yeah. Are there any questions about that warm-up? Okay. So let's go ahead then and start with the PowerPoint for today's lecture. Okay. So like I said, we are doing chi-squared tests today. So why do we use a chi-squared test? We use chi-squared tests when we're dealing with frequencies or counts, which means we're working with discrete data. And essentially what a chi-squared does is we're seeing if the expected values that the expected values, expected proportions, expected probabilities match what we actually saw when we ran our experiment or collected our data. There are three types of tests. There's the goodness of fit, independence, and homogeneity. These kind of work the same as the F distribution and the T distribution, where we compare our critical and calculated value. And based on the distance between those, we get a p-value. We also have the same statistical null and alternative hypotheses for each of those types of tests, the goodness of fit, homogeneity, and independence. The null is always that our expected or our observed values match our expected, and our alternative is always that they don't match, that our observed values do not match our expected values. However, there's different interpretations of our null and alternative hypotheses depending on the chi-squared test that we're running. For our goodness of fit test, we're testing to see if our observed values, our observations, follow a known distribution. So that's what's key about this one. It's a known distribution. For example, a binomial distribution, a Poisson distribution, Mendelian segregation or genetics, or a given probability. So essentially, we have an idea of what the observed values should be if they're following the distribution we expect them to be following. So we can kind of connect this back to when we were doing the binomial and Poisson. Remember, we were looking at that observed minus expected column to see how much deviation there was between our observed and expected values. Using a chi-squared test, we can actually calculate a p-value to see if those deviations are large enough for it to no longer fit a binomial or a Poisson distribution, or to tell us if the binomial or Poisson distribution or Mendelian genetics are not a good distribution to describe the data. As I said, for all chi-squared tests, observed and expected frequencies are always, our null and alternative hypotheses are always the same. However, the interpretation is a little bit different. So the null for the chi-squared goodness of fit test is that our observed frequency equals our expected frequency, but we interpret that meaning that it matches the distribution that we have expected. So it matches a binomial, Poisson, Mendelian, or given distribution. Our alternative is that the observed frequency does not equal the expected frequency, meaning that it does not match the expected distribution. Our data is not a good fit for a binomial distribution or a Poisson distribution or Mendelian genetics. Something else is going on, and something else is probably a better fit for the data. So here's some examples of what we could get using binomial Poisson and Mendelian genetics and the columns that we would be comparing. So for binomial, we've got the number of infected. Remember, this is from the example where we're looking at the number of infected insects or clams. I can't remember when we first did this chapter. So we have the number of infected per sample of 5. Remember, in this example, k equals 5. So we could have up to 5 successes per trial because we're pulling 5 bugs or clams and we're seeing how many are infected, which means that per sample of 5, 0, 1, 2, 3, 4, or 5 could be infected. We then have our observed frequency, this FI column. We have our expected proportion. We multiply our expected proportion by our sample size to get our absolute expected frequency. We have our observed minus our expected to get the deviation. So when we're doing a chi-squared, what we're really comparing is this observed frequency against this expected frequency. And how much difference is there between the two? How much deviation is there? Same thing with Poisson. We have the individuals per quadrat here from 0 to 8 and then greater than 8. We have the observed frequency, so how often have those occurred? Our expected proportion using the Poisson distribution, and our expected frequency, which is our proportion times our sample size. We're then comparing our observed frequency versus our expected frequency. What we actually got, how many we should have gotten if our data fit a binomial distribution. We haven't done Mendelian genetics or Mendelian segregation, but everybody should be familiar with Mendelian genetics. This is probably something you've taken in high school, biology, and other biology classes that you've taken in your undergraduate career. So hopefully we don't have to spend too much time here. Everyone should be pretty familiar with this. Just, I guess, a brief overview. So this is a dihybrid cross, or the results of a dihybrid cross. We have wild type, which would be, let's say we've got normal eyes versus curly wings. So let's see, we'll say normal eyes equals capital N, white eyes equals lowercase. We've got curly wings, or we've got normal wings. Let's do W, capital W. So normal wings, and then curly wings. Wild type would be a phenotype. So these are phenotypes. We're not looking, at genotypes. That's important to note. If we're looking at phenotype specifically, wild type would be dominant for both of these if we have complete dominance. So we'd have capital N. It doesn't matter what the second one is. It could be a capital N or a lowercase n for wings or for eye color, and then a capital W for having normal wing pattern. For normal eyes and curly wings, we would have a capital N, doesn't matter what this is, and then curly wings would mean we have 2 recessive. For white eyes and normal wings, that'd mean we'd have 2 alleles for the recessive white eyes, and it doesn't matter. We have at least 1 for normal wings. It doesn't matter what the second one is. It could be normal or curly. And then we have the homozygous recessive, where we have 2 pairs of recessive traits. Essentially, under these Mendelian genetics, if we're doing a dihybrid cross like that, we expect the ratio to be 9 to 3 to 3 to 1. We expect 9 out of 16 to be wild type, 3 out of 16 to be recessive in one trait but not both, and then 1 out of 16 to be recessive in both traits. So that's just kind of a quick crash course in Mendelian genetics to review. But essentially, so we have our observed number, We have our expected proportion. We multiply our expected proportion by the sum, our total number of samples, to get our expected numbers. And then we compare our observed frequency to our expected frequency. And we calculate a chi-squared calc based on those. So for these goodness of fit tests, you are always given observed frequencies, and you may have to calculate your expected frequencies. So keep this in mind when solving these problems. So here's a closer look at the binomial example. So we have our number infected, observed frequency, expected proportion, expected absolute frequency, our deviation. If you look in your lab menu, you also have this sign column where it's plus or minus, depending on whether it's above or below. In the chi-squared chapter, it's more just like the chi-squared contribution, which we calculate using this equation. It's for each index, our observed minus our expected, so our deviation, squared, and then divided by our expected. That gives us the chi-squared contribution for each of these. We then sum them together here to get our total chi-squared calc, our total deviation from expected. When we're solving these using the tables, the first thing we have to do is find our chi-squared critical value using degrees of freedom and alpha. For the goodness of fit test, the degrees of freedom is a little weird, depending on the distribution you're comparing against. So the general equation is this: degrees of freedom equals G minus 1 minus R. G is the number of groups your data has been split into, and R is the number of observations that you are estimating. So in binomial, we calculate X bar and K, and we actually have those. We have our sample average, our sample weighted average, and our trial size. We use X bar and K to estimate P. So we have one estimated parameter, R equals 1. For Poisson, we have our sample mean, our sample X bar. We use our sample X bar to estimate the population mean, which means that we also have one estimated parameter, R equals 1. For our normal distribution, we have our X bar, our sample mean, and we have our sample variance. And we're using those to estimate our population mean and population variance, which means R equals 2. It's a little bit easier to just remember this table here, though, instead of trying to figure out what R is for each of these distributions. And again, G is just the number of groups your data has been organized into. In this case, in this specific example, G is equal to 6 because we have split our data into 6 groups. We have 0, 1, 2, 3, 4, or 5 number of infected. That is 6 groups that we have split our data into. So our degrees of freedom for this problem would be 6 minus 1 minus 1. So our degrees of freedom is 4. We would then look that up in our table at the back of the lab manual. It's on page 279. And then compare that to our critical or our calculated value, which is the sum of these chi-squared contributions. So our observed minus expected squared divided by our expected, sum those up for each of these 6 groups, and we get our calculated value. We then compare our critical to calculated values. If our critical value is, if our calculated value is more extreme than our critical value, we reject the null hypothesis and say that our data does not fit the observed or the expected distribution. For our test of independence, we're testing to see if two traits are independent of one another. So we have two traits, are they independent? These could be eye color versus hair color. And these are always random sampling within one population. So I'm going to really try to point this out here. Keep this in mind because it gets a little tricky to tell the difference between independence and homogeneity. But in independence, you have one population and two traits. Again, our null and alternative are the same, but we interpret them differently. So again, it's our null is that our observed frequency equals our expected frequency. However, our interpretation is that the traits are independent of each other. Our alternative is that the observed frequency does not match our expected frequency, and we interpret that to mean that our traits are not independent. Our traits are dependent. So here we have a setup for an independence question. So we have eye color versus visual acuity. So how good is your vision versus what color are your eyes? The first thing we need to do is calculate the proportion of each trait. So calculating the proportion of brown, we would take the total number of brown divided by the total number of observations to get the proportion, to get the fraction of brown to all others. We would do the same thing for visual acuity. So we could take the number of good divided by the total number of samples to get the proportion of people that have good eyesight, regardless of eye color. We then use those proportions to calculate the expected proportion of obtaining each combination of traits. This is using the multiplicative rule of probability. Actually, this whole method should look very, very familiar from when we were doing probability and filling in these tables. The table is larger, but the way that it works is the same. So I know this one looks a little bit messy. That's because it has a couple different types of data within it, but what we're focusing on is the proportion we just calculated for good and for brown. If we're looking then what our expected proportion of good and brown is, if they're independent, that means we can use the multiplicative rule. That means this value times this value should give us what we expect the probability or proportion of good and brown to be if they're independent, using the multiplicative rule. We do that for each combination. We then calculate the expected number or expected frequency of each combination of trait from the expected proportion. So we take our expected proportion, this 0.09162, we multiply that by our total number of samples, and that gives us this number here. The expected frequency, the expected number of people we would expect out of 319 to have good vision and brown eyes. We do that for each combination to get the expected frequency. Finally, we calculate our chi-squared calc and our chi-squared crit and compare them to calculate a p-value. Again, we do that using this chi-squared contribution from each group. So we would calculate this chi-squared contribution for each of these groups, for this group, this group, this group, for each of those, and then sum them together to get our chi-squared calc. Which I have here. So you sum each of those contributions together to get your chi-squared calc. You then compare that to your critical value to get your p-value. For degrees of freedom, when looking up your critical value, it's actually a lot simpler. It's just the number of rows minus 1 times the number of columns minus 1. So here we've got 4 columns. So 4 minus 1. We've got 3 rows, so 3 minus 1. Our degrees of freedom for this problem is 3 times 2. So it's a lot simpler to look up or to find the degrees of freedom for homogeneity and independence compared to goodness of fit. Okay. And then we have our chi-square test for homogeneity. This is functionally the exact same thing as an independence test, but our interpretations are different again. And this time we're testing if one trait differs across multiple populations. So eye color across different ethnic groups. So we have different populations, different groups that we expect may have different frequencies and a single trait. Again, there's random sampling within the multiple populations, but this is really key. There's one trait and 2 populations. Again, null and alternative are the same. Observed equals expected. Observed doesn't equal expected. But again, interpretations are different. For homogeneity, the null hypothesis means that our traits are homogeneous across populations, which means they're evenly distributed or equally distributed between populations. Our alternative is then that the traits are not homogeneous across populations. Our traits are not evenly distributed between populations. So let's look at this example. This looks almost identical to the one that we just looked at for independence, but now we have sex. We have male versus female, and we have visual acuity. So we expect under the null hypothesis for these numbers to be roughly equal to each other. For the number of males and females that have each of these visual levels to be approximately equal. And we see that here when we look at these proportions, right? So we've got 200 people. We would expect that we would find a 50-50 split. And then we would expect, we would then calculate our proportion of visual acuity between these two using the total of each divided by our sample size. So using those proportions, we then calculate the expected proportion of obtaining each combination. So again, the same thing that we just did for independence. We then calculate our expected number of each combination of traits from the expected proportion. And then we calculate our chi-squared calc and chi-squared crit and compare. These slides should look similar because they're exactly the same slides from independence. And just like with independence, our degrees of freedom is our number of rows times our number of columns. So here we've got 3 columns minus 1, and we've got 2 rows minus 1. So our degrees of freedom is equal to 2 times 1. Or alternatively, we could do all of this in R. The way that these slides have been broken down, break down each of the steps of solving the chi-squared test, but thinking it through in that way is really how you would do it in Excel, which the lab manual walks through in a lot of detail. But again, we're going to focus on R, and it's pretty quick to do in R as well. There's only a couple lines of code that you need to know. Doing this in R, we get an exact p-value. So again, same as always, if our p-value is less than 0.05, we reject the null hypothesis and accept the alternative. If our p-value is greater than 0.05, we accept the null hypothesis and reject the alternative. Your lab manual also mentions that there are 2 corrections that can be applied to the test to make your results more accurate. These are the Yates correction and the Fisher's exact test. We do these when we have really small sample sizes or when we're doing a 2x2 chi-square test. So if our test looks like this, like we have, I don't know, one group versus another group, and I don't know, I'm just picking letters, and we have counts here. These are more common in medicine, I think, and in clinical studies. If we were doing something like this, that's when you would apply these corrections. I'm just going to tell you now, though, that I'm not going to do many of these, so you don't have to worry, for lab at least, you don't have to worry about doing these. The R code varies a little bit between whether we're doing goodness of fit or independence and homogeneity. For goodness of fit, we actually create something called a vector, which I'll explain more when we get to using R. But essentially, a vector is just a string of numbers or characters or values. It's not an actual data set. When we talk about a data set or a data frame, as they're often called, we're talking about having a 2-dimensional set of data where we have rows and columns. We have typically our observation number here, so our index, so 1, 2, 3, 4, 5, so on and so forth. And then we have maybe like eye color, ethnicity, I don't know, sex, vision, like that, where we have different columns up top. And then we have values here, so like brown, hazel, blue, ethnicity, I don't know, black, white, I don't know, so on and so forth, right? So our columns go down this way, and across is just an individual person. So this would be one person is black, has brown eyes, I don't know, is female, has great vision, I don't know. When we have vectors, though, it's literally just a string of numbers. So that's the big difference between a vector and a data frame. We then have to create, so we have to create 2 vectors. We have to create a vector for our observed frequency and our expected proportions. So this is important too, our expected proportions. And the reason for that is the chi-squared test function. What goes inside of that is our expected frequency or observed frequency and our expected proportion. We have to use this argument where it's this p equals, and this p just means probability or proportion. We're saying, hey, R, do a chi-square test, compare our observed frequency to this expected proportion. Our probability or proportion equals this. For independence and homogeneity, we actually have to create something called a matrix. And this is similar to a data frame, but functionally works differently in R. So it actually looks a little bit more similar to what we're used to seeing in Excel. Whereas instead of having index over here and attributes up at the top, what we have is we have, you know, like sex over here, and we've got female, male, and then vision up top. We've got great, good, poor. So now we've actually got we've got columns across, or I should say categories by row and by column, and then we have numbers in the center that represent the intersection of those. So this is a female with great vision. This is a male with good vision here. So we make a matrix. For these matrices, you actually import data. So we don't type out data like this. You will import data using the same method that you did for using the t-test. So file.choose, read.csv, pull in your CSV. We use this function called xtabs, which makes our matrix. And then we create an object containing the matrix. We then just do the chi-square test on the name of the matrix that we just created. Okay. So we can actually, let's just go ahead and do all of that in R. I think it'll make a little bit more sense. Before we start with that, are there any questions about the chi-squared tests? There's a bunch of stuff that's in the lab manual that we are not covering. Let me just have a list of it that I want to mention that we are skipping. Okay, so we are skipping so yeah, we're generally skipping the Excel calculations. We're skipping the corrections. We're not doing one-tailed chi-squared tests. We're just doing two-tailed chi-squared tests. So similar to how we did it with the f-test. We're only looking, do our observed and our expected values match? Are they the same? We're not saying our expected values for one group bigger or smaller than the other. So it's all just a two-tailed test. What else are we skipping? Yeah, the small expected frequencies section, we're not doing that in lab. And I believe that's it. Okay. So it doesn't look like there are any questions. So let's switch gears to working in RStudio. Okay. So this is the same, set up the same way as everything that we did last week. So I've got everything broken up for you. You can click in this little outline area to skip through things. I actually don't have everything uploaded to the cloud. So let's upload these examples really quick. And you'll notice that unfortunately you can't upload multi-files. I think you can, but you have to create a zipped file first, which is, fine if you want to go ahead and do that. It's not a bad idea at all. Okay, so now we've got everything loaded in. So because goodness of fit and homogeneity/independence function differently, and they test things a little bit differently, when we do a goodness of fit versus doing independence or homogeneity, the input is a little bit different, as I kind of mentioned in the PowerPoint. So make sure that you're using the right code, the right functions based on the test that you're doing. The main functions that we're using are chi-squared test and xtabs. Those are the two new functions. You've seen everything else. So when we do a goodness of fit test, we do not load in an associated CSV. Instead, we create vectors. And again, these vectors are just strings of data. They're just lists of numbers or characters. They're not actual data frames or structured two-dimensional data sets. So when we do these goodness of fit tests, I have it written out in some steps right here. We've got number one, we need to create a vector for observed values, which you'll get from the problem. And we do that with this line right here. So observed, we're creating an object called observed. We're putting inside of it, we're assigning inside of it using this arrow. And then I suppose this is actually a new function as well. This C stands for concatenate or combined. And we are combining or concatenating a series of numbers separated by commas. So we're putting that series of numbers inside of this new object called observed or OBS. The second step is to then create a vector for the expected values or expected probabilities for each event. And we do that the exact same way. So we have our expected probabilities. We're assigning inside of it this series of numbers using this arrow. So concatenating our proportions by commas, separated by commas. Just a general tip, super important that you remember to do this. Make sure that you create unique names for things in R, because if you don't, R will just override the data and you'll lose all of the old values. So today we're going to be doing a lot of chi-squared tests, right? If you name every one observed and expected, you're overriding previous observed and expected objects. So we want to create slightly unique names. So if we're doing, or you'll see down here for observed and expected, I have example 1, period, OBS, example 1, expected. So I know these are for example 1, this is the observed values, and these are the expected values. We want to do that with everything we create in Excel, or not in Excel, in R. Okay. So there's an example of a goodness of fit test on page 153. We're looking to see whether observed traits in a sample of Drosophila follows the expected Mendelian genetics distribution as predicted by the Punnett square. First, let's go ahead and write out our null and alternative hypotheses. I have already gotten them written out here. So our observed equals our expected frequency is our null hypothesis. Our alternative is that they are not equal. Our observed frequency does not equal our expected frequency. Remember, those are always the hypotheses for doing these chi-squared tests. However, the interpretations vary. So the null hypothesis for goodness of fit is that the observed frequencies of phenotypes will follow the Mendelian distribution. So that's specific to this problem, and I want you to be specific for each problem. The observed frequencies of phenotypes will follow the Mendelian distribution. The alternative is then that the observed frequencies of phenotypes will not follow the Mendelian distribution. So let's go ahead and test that. We're going to create our observed frequencies object and our expected proportions object. And again, you can copy these, but I recommend you type them out. Get this in your muscle memory so you know what you're doing when you have to solve exam problems. So EX1 for example 1, OBS for observations. I'm creating this arrow because I'm putting stuff inside of that. I'm creating a new object. C for combine or concatenate, open parenthesis, and I'm going to type the expected values. And again, these are on page 153. The whole written out word problem is on page 153. And I believe, yeah, on page 153, there's a whole Punnett square written out. explaining how this dihybrid cross functions. So what we actually observe is we get 180 of these wild type flies, 143 of normal eye/curly wings. We get 126 of these white eyes/normal wings, and 51 of these white-eyed curly wings. I'm going to run this line, holding Control or Command and hitting Enter. And when I look over in my global environment, I see now that I have this new object, example 1 observations, and it's a series of 4 numbers. You'll notice that it doesn't show up under data. Data is where matrices and data frames go, these two-dimensional data sets, right? If I click on warm up, I see it. It actually pops up as a new tab over here. And I see we've got observation and then columns across. So we've got two-dimensional data. That goes under data. Vectors, vector strings, go under these values. Okay, so now let's make our expected proportions. So we got example 1, EX1, EXP. Example 1, expected proportions. Using the arrow to put these numbers inside of our new object, C for concatenate, and let's type in our expected frequencies or expected proportions. Following a dihybrid Mendelian segregation genetics expectations, we should get a ratio of 9:3 to 3:1 for these, which means 9 out of 16, that's the proportion, right? 9 out of 16 should be wild type, should show the dominant phenotype for both traits. 3 out of 16 should show 1 recessive and 1 dominant phenotype, either one, so either normal eyes, curly wings, or white eyes, normal wings, right? In both those cases, normal eyes is dominant, curly eyes is recessive, white eyes is recessive, and then normal wings are dominant. So 1 dominant trait, 1 recessive trait, 3/16 is the frequency or the proportion we expect for both of those. And then we expect that 1 out of 16 flies should be homozygous recessive, should show completely recessive traits. So let's type that out. 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16. And you'll notice that these correspond. It's very important that these go in the same order. We expect 9 out of 16. That's the expected proportion for this 180, for this wild type. 3 out of 16 is the expected proportion for this 143 and 126. 1 out of 16 is the expected proportion for this 51. So it's really important that we keep these in the same order. I'm going to run this line now. So Control or Command, Enter. R translated those into decimals, which is totally fine. So we expect a little bit over half of them to be wild type, 0.18, so a little less than a fifth of them, which makes sense, right? 3 out of 16. So a little bit less than a fifth of them to have 1 recessive and 1 dominant trait. And it doesn't show, but 1 out of 16 to be 51. So now that we have our 2 objects, we can run our chi-squared test. And the function for that is pretty straightforward. It's chi-squared.test. We will always make an object out of our chi-squared test so that we can pull additional information out of, which I'll explain in a second. And same with when we made these objects up here, we didn't just do observed and expected, we did example 1 observed, example 1 expected. We're going to do the same thing with the chi-squared test. We're going to do example 1.chi so that we know what chi-squared test we're looking at. So let's go ahead and do that. This is the general code for doing a chi-squared test. Name.chi, so specific name, example 1 in this case. We're creating an object, so we're using this arrow. Chi-squared.test. Our observations for that problem, so this would be example 1 observations, comma, the proportion or probability equals, and then the expected probability or proportion. Right here is the code that we're going to type inside of this code chunk to do the chi-squared test for this problem. So example 1, chi-squared, that's what we're naming our object, chi-squared.test. Example 1 dot observations. So we're comparing our observed frequencies for example 1 against the probability, and you can put spaces, you don't have to, it doesn't really matter, of the expected proportion or expected probabilities for example 1. Now, control or command, enter. We don't get any output here, but if we look over here, we now have data. So it didn't show up in the values, it showed up in this data area. And it's a list of 9 pieces of information. Now, to actually look at what the results of our chi-square test are, we're going to need to print that. We're going to need to print our object, to view our object. So we're just going to type example1.chi and run that. And we get our chi-squared results here. I want to also point out, when you start typing this, when you look at this list here, these little symbols tell us stuff. So this example 1, example 2, those tell us that those are like lists of numbers. Those are data, right? This one has this funky little thing here. We've got like this, like these 3 squares or these 3 rectangles that are like kind of like in a circle. That tells us this is actually a model. This is a model. These gives us the results of some sort of specific test or model. That's what that means. So when we print this, again, we run that line, we get this chi-squared test for given probabilities. We get what data we used. We get our chi-squared calc, our degrees of freedom, and our p-value. I'll come back to this. I'm pretty sure that we need to correct what the degrees of freedom here is. But we'll keep moving on. I'll come back to this and we'll make that correction in a bit. But we do get our chi-squared calc, our degrees of freedom, and our p-value. So statistically and biologically, we can come to some conclusions. Statistically, our p-value is less than 0.05, which means we can reject our null hypothesis. Biologically, what that means is that our observed distribution of phenotypes does not match the expected Mendelian distribution. We probably have some sort of other genetic mechanism going on here. It's not just a regular cut and dry dominance versus recessive inheritance. We're not quite finished though. So we've got one other thing that we can actually do because we created this object with our chi-squared test. We can actually pull out additional information, including our expected frequencies. So when we type out this, so example 1 chi-squared, our model, our chi-squared test, if I put a $, I actually get this whole list of different information that I can pull out of it. If I did statistic, that just gives us our chi-squared test or our chi-squared calc, which we got out here. So for the most part, the things that we can get out of this chi-squared test function were actually already printed for us when we printed the chi-squared test. Things that were not, though, include these expected frequencies, residuals, and the standard deviation of the residuals. We don't need to really worry about these. We're going to look at these expected frequencies, though. So Control Enter, and we get this string of numbers. These are the number of observations we should have gotten for each combination of phenotypes, for each phenotype, assuming that our data followed a traditional Mendelian distribution. It's a little annoying. We can like scroll back up and kind of look at our original data. We could also just print them together. So we could type example1.chi $ observed. We can actually run these lines together. So, because if I ran this by itself, it just replaces it. But if I want to look at these side by side, I can run these lines at the same time. I can do this by highlighting both lines and then doing Control Enter. I can also, just while the blinker is highlighted anywhere in this code chunk, press Control, hold Shift, and then hit Enter. And R will run both of the lines of code together. So now I can directly compare my expected and observed values. Just looking at this, I can see that there were way fewer observed than we should have gotten. There were way more of these, or sorry, there were way fewer of these wild type than we should have gotten. There's almost 100 fewer. We can see that there's a lot more of these one dominant, one hetero, or one dominant, one recessive trait observations than we should have gotten. We've got 143 and 146 compared to 93.75. And we see that we actually observed a lot more of these homozygous recessive than we should have under the assumption of Mendelian distribution. If we wanted to get a really precise look at that, we could actually have just done example1 chi observed minus our example1 chi expected. When we run this line, so Control Enter, it actually just subtracts those numbers. Our observed was, for the wild type, was under by 100. Our observed for the two middling traits were over by 49 and 32, and our homozygous recessive was over by about 20 observations. So we do this so that we can actually visualize what categories are driving our unexpected distribution or our deviation from our distribution. In your assignments, I will probably ask you to explain what seems to be driving these differences. Where is the deviation occurring? So that's what you would say. You would say we have about 100 observations fewer for the wild type, about 49, 32 observations more for the one dominant, one recessive trait, and 20 observations more for the recessive trait. That's why it's not fitting this Mendelian distribution. For independence and homogeneity, we are actually going to create a matrix using data that we upload from the CSV. And I wanted to make a note that the code that we're going to use for these problems is different from the code in the lab manual. We've found that this code is more consistent with later chapters, making it easier to learn how to do later analyses. So we're trying to keep things consistent for later chapters. If you read through the lab manual and you notice these differences, that's why we're not using the code that is given to us in the lab manual. So we are going to create a matrix from the data that we've imported, that we've got down here. Similar to how we did everything before, we named the chi-squared test example1 chi. We're going to keep the same good coding practices when we make matrices. We're going to have the specific name in front, dot, and then matrix at the end. So we know what problem we're working on and that it's a matrix. Xtabs is the function that we're going to use to create these matrices. And what goes inside of it is this. So we've got the name of our matrix, we're creating an object, we're using Xtabs to do it, and this is what goes inside. We have the counts, so the number of observations we saw. We're organizing that by, that's what this tilde means, organizing by category 1 and plus category 2. We then put the name, data equals, and the name of our datasheet object. And I've got that written out here. So after we make the matrix, we then just do the chi-square test on the matrix. So we don't have to do the expected, comma, p equals observed. We just put our matrix inside. And then we look at our results. So on page 154, there's an example for independence. We are working with a chi-squared independence test. We are testing to see whether two traits are independent in a single population. That's the difference between independence and homogeneity. For the example 2 on page 154, we are testing to see if the trait eye color is independent from visual acuity, so how well you can see. Our null hypothesis, again, is just observed frequency equals expected frequency, but specifically, we're saying eye color and visual acuity are independent of one another. Our alternative is that our observed frequency does not match our expected frequency. Specifically, that means that our eye color and visual acuity are not independent of each other. So hopefully you've already downloaded all of the CSVs and have uploaded them into your RStudio Cloud. If you haven't, go ahead and do that. And we're going to load our files in using file.choose. Again, if you're using RStudio, you don't have to do the other step. You just start here at this file.choose. So I'm going to run this line, Control, Enter, and select, I believe it is this one, this chapter 14, example 2 CSV. So that's the file location. I'm going to select the entire thing, Control C to copy it, click inside these parentheses, Control or Command V to paste it, Control Enter to run that line. I now have an object called chapter 14.example2. Let's check to make sure that it uploaded right and to see what our column names are. So Control Enter on this headline. And we have count, eye color, and vision. So we had 40 people with brown eyes and good vision, 27 people with brown eyes and moderate vision, 22 people with brown eyes and poor vision, 20 people with black eyes and good vision, 12 people with black eyes, moderate, so on and so forth, okay? So now we can create our matrix. And we're going to input this line and this line into this code chunk. And again, remember that R is case sensitive, and looking at the output from our head function, we see that the count is capitalized, eye color are capitalized, vision is capitalized. So our example 2 matrix is what we're making a new object. That's why we need this arrow. We're using the Xtabs function to do it. It's count tilde, which is up in the top left-hand corner of your keyboard, right underneath of your escape key. We're organizing count by, that's what that tilde means, organizing count by vision and plus eye color. And it doesn't matter if you do eye color and then vision, vision, then eye color. That it doesn't matter. comma, data equals chapter 14 dot example 2. That's the data that we're using to create this. Control, enter. And I see that we've got a new matrix over here and values, example 2 matrix. Let's then view our matrix. So example 2.matrix, control, enter, and we can see our matrix below. So we've now transformed this data in what's called long format, because it's a long list, into this wide matrix format. So we've organized our count numbers by eye color and vision. Now we can do our chi-squared test and view the results. So let's save our chi-squared test as an object though, remember? So we're going to do example 2 chi, because we're working on example 2, we're saving a chi-squared test, chi-squared test, and we're just going to put the matrix inside of these parentheses. So example 2 dot matrix. You can run that line there and create it, or you can just keep going and type your example 2 chi and run those lines together. It doesn't matter if you run one and then the other, or run both of them at the same time. However, if you try to run this line before running the line above it, you'll get an error because it doesn't exist yet. So I just ran those together and I got the output of our Pearson's chi-squared test. We get our chi-squared calculated value, our degrees of freedom, and our p-value. So we can conclude that we reject the null hypothesis because our p-value is less than 0.05. Biologically, that means eye color and visual acuity are not independent. Again, we can look at our expected values to see if our, we can look at our expected values if our traits were independent. So example 2 chi, $, expected, tab, control, enter. And these are the numbers we should have gotten if eye color and vision were independent of each other. We could look at this and then scroll up to compare here, but again, if I want to look at them at the same time and compare them side by side, I'm going to do that right here. So chi-squared observed, example 2 chi-squared, $, expected. Remember, I'm just using the arrow keys to go down and tab to select it. I'm going to run these lines together so I can view them side by side. So control or command plus shift and then enter. And now I can compare them side by side to try to figure out where the deviation occurred. It looks like there's a little bit of strange deviation going on in this blue column. These ones, there's some deviation, but they match a little bit better. We've got some weirdness, I think, kind of going on in this good column and in this blue column. We can look a little bit more closely at that if we want to subtract them. So let's try doing that. And I think I've already got this typed out for you. So control, enter. So where are the biggest deviations occurring? So we've got a lot of kind of 1-point stuff, 3-point stuff, but we can see the biggest deviation is occurring at this blue moderate, blue good, brown, right here. These kind of ones are driving this difference. So it looks like maybe we have too many people who have blue and moderate eyesight, not enough people who have blue and good eyesight, too many people that have brown eyes and good eyesight, and too few people maybe that have brown eyes and poor eyesight. So you can pull out a couple of patterns here. So where are the biggest deviations? Too many observations of people with blue eyes and moderate vision. Too few observations of people with blue eyes and good vision, and say too many observations of people with brown eyes and good vision. So those are 3 big deviations that I see here. There's an example of homogeneity on page 153 or 55. And remember, the way that we do independence and homogeneity is the same, but the interpretations are different. So we're asking, is a single trait homogeneous between 2 populations? So on page 155, we are seeing whether visual acuity is homogeneous between females and males and females. So let's write out our null and alternative hypothesis. So our null, observed equals expected. Alternative, observed does not equal expected. Again, interpretations vary though. So specifically, visual acuity is homogeneously distributed between males and females. That's our null hypothesis. Our alternative is that visual acuity is not homogeneously distributed between males and females. So there's some sort of unequal distribution between males and females and visual acuity. So this one has a different data set, so we need to make sure that we choose and read in this new collection of data. So file choose. This is example 3, so chapter 14, example 3, CSV. Open. Select my file pathway, Control C to copy it, paste it in these parentheses for read.csv, Control V, Control Enter to run that line. I see that it shows up over here now, chapter 14, example 3, and then run the head to view the data. Oh, it looks like these columns are actually backwards. So we have count, sex, which is not this, vision, male, female. So column names are backwards, unfortunately. That's not going to have any consequences though for the p-value calculated. If this bothers you, you could actually open up your CSV, switch the column names, and then save it and re-upload it. But like I said, it's not going to actually change the outcome of these results. So let's go ahead and create our matrix. So example 3, matrix. It's the object we're creating using X tabs, organizing count. Count by vision and sex. Our data is equal to the chapter 14 example 3 object we just created. So selecting example 3, tab, Control Enter, and you should now have an example 3 matrix under your values. So now let's create and view the chi-squared object. So let's create the object. So example 3.chi arrow, because we're making an object, and running the chi-squared test. We don't have to do the expected, comma, p equals. This time we just put our matrix in. So example 3.matrix. Tab, Control Enter. I just made the chi-squared object. Now let's look at what's inside. Let's look at the results. So example3.chi. Control Enter. And we get the output. We get our chi-squared calc, degrees of freedom, and our p-value. So we are going to accept the null hypothesis because our p-value is greater than 0.05. Biologically, that means visual acuity is homogeneously distributed between males and females. Okay, so we've got another class data collection activity. Hopefully a lot of you already know what your Hogwarts house is. If not, you're gonna actually take this BuzzFeed quiz. It's pretty quick to figure out what your house is. So imagine we're all at Hogwarts and we are all preparing to be placed in our appropriate houses. It is apparently spooky season after all, so I feel like this question or this activity is pretty appropriate. So we could have Gryffindor, Slytherin, Hufflepuff, or Ravenclaw as our Hogwarts houses. You hypothesize that students will be equally distributed across all 4 houses. On the Excel sheet your TA shares with you, that's me, I will give you the sheet, indicate which house you belong to and your biological sex, male or female. If you don't know which house you belong to, go ahead and take this quiz. And I will share with you the Excel sheet to put your information in. I got pretty much an even split between Gryffindor and Ravenclaw, but I think I am slightly more Ravenclaw according to the test, but oops, that's not where I meant to put that. Okay, so the link for the Google Sheets is in the chat. So go ahead and fill out your results there, or go ahead and take the quiz and then put your results in. So what test are we going to run? Which of the 3 types of chi-square tests are we going to run on this data? Are we doing goodness of fit? Are we doing independence? Are we doing homogeneity? Yeah, homogeneity. So it's homogeneity because we're looking at a single trait between 2 populations. A lot of the time for problems like this, and I'll keep it consistent as well because this matches up with how the lab manual does it, sex, male, female, are considered 2 separate populations, I think because we expect that males and females have different attributes, I guess. I don't know. I think it's kind of a weird system, but yeah, so we'll count sex as 2 separate populations. We're looking at 1 trait, and that trait is house. So we're doing a test of homogeneity. What are our hypotheses? In the chat or unmute yourself, either way. Well, I guess first, right, our null and alternative is always that our observed frequency equals our expected frequency. And our alternative is that our observed frequency does not match our expected frequency. But specifically for this problem, for homogeneity, what are we expecting to see? Yeah, exactly. That Hogwarts houses is homogeneously distributed between males and females. We expect to see that there is no difference in distribution between males and females. Alternative is that they are not evenly distributed. Okay, so hopefully you've already uploaded your CSV. Let's go ahead and select it and run the data. So it is this Bio260 Hogwarts Houses 09b.csv. Going to select it, copy, Control V to paste it inside this Hogwarts object. And then I didn't type Hogwarts in here, but Hogwarts will go inside the head function, Control Enter to view it. And there we go. We've got the number of people that were in each house and their sex. We can then make a matrix out of this data. So we're going to call it Hogwarts matrix. Making this an object, so we need this arrow, X tabs. We are organizing counts by house and sex. The data we're using is Hogwarts. That's the object that we created that has the data inside. The Hogwarts matrix. Wait, no, the Hogwarts matrix is what we're making. So yeah, just Hogwarts. Control Enter. And now we see it in our values. Let's run our analysis and, but first when we do that, we need to make our chi-squared object. So Hogwarts chi, using the chi-squared test on our matrix. So Hogwarts dot matrix. Hogwarts dot chi. So I haven't run either of those lines. I'm going to go ahead and run them together. So Control Shift Enter. It tells us our chi-squared approximation may be incorrect. So it's actually giving us a warning that the degrees of freedom may be incorrect, although I believe, oh, it's giving us this warning because there are very few observations. So this is actually a case when we would do a correction for too few observations. Like I said, though, we're not going to worry about that for this class. So we'll just go with this for what we have. Yeah, because I think if we just printed, yeah, it's because we have all of these ones. We have these single observations for a couple of things. R is telling us that it might be less accurate because the sample size is so small. So that's the warning that R is trying to give us right now. But we've got our chi-squared calculated value, our degrees of freedom, and our p-value. So what are our results? Statistically, do we accept or reject, and why? Yeah, we accept the null hypothesis because our p-value is greater than 0.05. Biologically, relating this back to the actual problem, that means Hogwarts houses are evenly distributed across sex. Cool. So good job. Let's see. Do you want me to work through another problem with you before putting you in the breakout groups? Okay. So let's see. Everyone pretty much said yes. Angelina asked for number 2, so sure, because I think a lot of you probably started on number 1. So let's go over number 2. You are studying a flower species that is only purple or yellow. The color gene is controlled by gene P. Purple, capital P, is dominant over yellow, lowercase p. When you cross 2 heterozygous individuals, you get the following results: 23 purple and 7 yellow offspring. Do these color traits follow the expected Mendelian distribution predictions? So this is a goodness of fit test, right? We're not testing if a trait is evenly distributed between 2 populations. We're not seeing if 2 traits are independent of each other. We have a predicted distribution, a Mendelian distribution, and we're saying, do our observed frequencies match our expected distribution? So because this is a goodness of fit test, we don't need to import any files. We need to make vectors. Let's see, just another quick recap on Mendelian distribution. So, okay. So we've got flowers, and they are either purple or lowercase p yellow. So this purple is dominant over this. So if we take if we do a test cross or a hybrid, a hybrid cross between heterozygous individuals. So we've got flower 1 that means flower 1's genotype is capital P lowercase p. Flower 2 has the same genotype, capital P lowercase p. When we do a Punnett square, which you all should know how to do, when we make this Punnett square, this is the breakdown that we get. We get big P, 1 big P, 1 big, 1 little, or two big, sorry, 2 with 1 big and 1 little, and then 1 with 2 littles. So our genotypic distribution, which relates to our alleles, is capital P, so 1 capital P, 2 big P, little p, and 1 little p. So we get a 1 to 2 to 1 ratio. In other words, 1/4 versus 2 out of 4 versus 1 out of 4. It's the breakdown of the ratios that we get for these. If we're doing a phenotypic ratio, which is what this problem is asking, actually, let's see. So our phenotype is just purple versus yellow. So phenotype is what you see. Purple would be this, or this because capital P is dominant. So it doesn't actually matter. It's just capital P and then whatever the second allele is, it doesn't even matter. Versus yellow, which is just lowercase. So we would see 3 purple for every 1 yellow. Our ratio is 3 to 1. In other words, 3/4 and 1/4. So those are our expected frequencies, or our expected proportions, 3/4 and 1/4. Okay. So there's no file import needed, but we do need to make those vectors. What test are we running? This is a goodness of fit or just a GOF chi-squared test. Our hypotheses are always, remember, observed frequency equals expected frequency, or our observed frequency does not equal our expected frequency for the alternative. But specifically, what we are actually testing in this problem is whether or not our data fit the Mendelian distribution. So our null hypothesis is flower phenotypes match Mendelian distribution. So a 3-to-1 ratio. Our alternative is that flower phenotypes do not match Mendelian distribution. So they do not match this 3-to-1. To do the test, we need to create those vectors and then run the chi-squared test. So let's see, what question? Question 2. So I'm going to do question 2, observed values. So I'm creating an object called question 2 observed. I'm using this C to concatenate, to combine values. The numbers are given to me for the observations. We have 23 purple and 7 yellow. So 23 and 7. I'm now going to create the expected the expected proportion. So Q2 expected proportions. Creating an object, so I use this arrow. C to combine. The proportions, remember, are 3 out of 4 and 1 out of 4, or 0.75 and 0.25. I haven't ran either of these lines yet. I'm going to do Control-Shift-Enter to run them both. I now see that I have expected, my Q2 expected 0.75, 0.25, my observed 23 and 7. So right here, remember, you can make notes inside of your R code using the hash key. So making the objects for observed and expected. The next step is to do the chi-squared test. We want to make an object when we do this. So I'm going to do Q2 chi-squared is the object I'm making. I'm using the chi-squared function, chi-squared test function. The input for this is Q2.observe. So your observed value, comma, P equals, our proportion or probability equals the expected proportion. Control-Enter. I see that up here. So that ran fine. Q2.chi. When I run this, I get the output. So we've got our chi-squared test, our degrees of freedom, and our p-value. Conclusions then? Statistically, do we accept or reject? Accept, yeah. Accept the null p is greater than 0.05. Biologically, what does that mean? Yes. Yes, it means that it matches the Mendelian distribution. Our data matches this 3-to-1 ratio. Flower phenotypes match Mendelian distribution. And then I ask you, are there any patterns? If we print our original data, so Q2 chi, observed, Q2 chi, expected, this is what I'm talking about. I want you to tell me, is there anywhere where we see like a strong deviation? In this case, not really. We actually see that there's only a 0.5 deviation here and a 0.5 deviation here. This data almost perfectly fits this Mendelian distribution. And this would be impossible to get because we can't have counts of 22.5 purple and 7.5 yellow. So I would honestly say no. The data the data fits the distribution almost perfectly. Oh, okay. So let's run through these practice problems. For question 1, you're interested in the color blindness trait among males and females. Males can only be colorblind or have normal vision, while females can be colorblind, have normal vision, or be carriers of the color blindness gene. You are interested in color blindness trait frequency among these two groups. So we're going to have to look at our CSV and import that first. Okay, so let's do the file.choose to select our file. This is the chapter 14 question 1 CSV. So open, I got my file pathway, Control C to copy, Control V to post it in the read.csv function, Control Enter to run it. We need to type it into this head function. So chapter 14.q1. Control Enter. And there we have our count, so the number of people for each sex, and then a trait of color blindness. Colorblind, carrier, not colorblind. The test that we're going to want to run for this is the chi-square test for homogeneity. So female and male are considered two populations. So we're doing a test of homogeneity. Our null is that our observed frequency equals our expected frequency. More specifically, that color blindness is homogeneous between males and females. Our alternative is that our observed frequency does not equal our expected frequency, and color blindness is not homogeneous between males and females. So let's make our matrix and do the test. So our matrix is going to be, I'm going to call it q1.matrix. And I'm going to use X tabs to make it. Let's look at our data again. I'm going to organize count by sex and trait. Data is equal to the matrix, our Q, what is it? What did I call this data? Chapter 14, Q1. That's the object, it's it. Chapter 14, Q1. Control Enter. And now it is over here. Let's print that out and see what we got as our results. Oh, those are our matrix, sorry. We still need to make the chi-squared. And we do that doing q1.chi, so we're making an object, chi-squared test, and we're putting our matrix inside it. So our q1 matrix. And now let's view the results of that, q1.chi. And we get a chi-squared calc, our degrees of freedom, and our super, super small p-value. So statistically, we are going to reject the null. P is less than 0.05. Biologically, that means traits of color blindness are not homogeneous between males and females. And then I ask you, are there any patterns? To view the patterns, you could do it up here, or you could actually create a new chunk. Remember, Control Alt I to insert a new chunk. Let's see, what is this? Q1 chi. I'm going to look at the observed Q1 chi. I'm going to look at the expected. I'm going to run those two lines together. Control, Shift, Enter. And looking at this, because it's homogeneity, we expect the frequency of these to be equal to each other. So we expect them to be evenly distributed. Which is why we see this kind of even split between these. Looking at this, I can say, let's see, the biggest deviations are probably that there are no male carriers, for one, right? We have 0 male carriers. We expected there to be 10. There is also very few. There are very few females that are colorblind. Too many males that are colorblind. And too many females that are carriers. So those are the biggest deviations. We could also look at that by subtracting the 2. So Q1 chi observed minus Q1 chi expected. And we get those written out as numbers. So too many female carriers, too few male carriers, none really. Too few females are colorblind, too many males are. And then the deviation between not colorblindness is actually pretty small, only about 2 people off for each. For question 2, we are studying a flower species that is only purple or yellow. We already did this one in class, so actually I'm not going to go over it again. We did a goodness of fit. Phenotypically, we would expect a 75%, 25% split, and our data pretty much perfectly fits that. For question 3, you've explored your curiosity about phenotypic ratios of the flower offspring. Now you are interested in the genotypic ratio of the flower offspring. Remember, you are crossing 2 heterozygous individuals. After you run a genetic test on all your flower offspring, you get the following results: 8 homozygous dominant, 15 heterozygotes, and 7 homozygous recessive. Does your genetic data follow the Mendelian distribution you predicted? We don't need to import any data here because this is another goodness of fit test. So goodness of fit. Our hypotheses are observed frequency equals our expected frequency. Our observed frequency does not fit our expected frequency. Specifically, our genotypes fit Mendelian distribution, and our genotypes do not fit Mendelian distribution. let's go ahead and do that test. We need to create our objects. So this is Q3 observed values. I'm concatenating or combining those numbers. So what do you see? We have 8 homozygous dominants, 15 heterozygotes, and 7 homozygous recessive. Q3 expected proportions. Our expected proportions when we have a hybrid cross, a heterozygote cross like this, is 1 to 2 to 1. So 1 out of 4 would be homozygous dominant, 1/2 would be heterozygotes, and 1/4 would be homozygous recessive. We can now do the test. I haven't run these lines, but that's okay. I'm going to actually type everything out and then run it all at once. So after I make those, I would do my chi-squared test. So let's see, Q3 chi, saving it as an object, chi-squared test. I would be doing it on Q3 observed, comma, P equals Q3 expected. And you'll notice that now these look a little bit different, right? We have these yellow boxes. Those yellow boxes tell me that R recognized that these are objects that don't exist yet, but according to the code that I have typed here, I'm creating them. So it's still saying like these don't exist yet, just a warning, but it looks like you're trying to make them. So going with expected. I'm then going to print that object. And I'm going to go ahead and look at the chi-squared expected, chi-squared observed. And I'm going to actually calculate the difference between the two of them. So Q3 chi-squared observed minus Q3 chi expected. And like I said, I haven't actually run any of this yet, but I already know what I'm going to need to type out. So I've just typed it all. And now I'm going to run all of these lines at the same time. Control, Shift, Enter. And it created these objects, created this object, ran the test, printed the test, printed out my expected and observed, and then below it, the difference between the two of them. So you can definitely do this. Just be aware that it's not going to tell you exactly what they are. You kind of have to remember what you asked for. So I know this is expected because it's the first thing I asked for. I know this is observed because it's the second thing I asked for. And I know this is the difference because it's the third thing I asked for. So our conclusion, statistically, we're going to accept the null hypothesis. Because our p-value is greater than 0.05, it's almost 1. Biologically, that means our flower genotypes match a Mendelian distribution. Any patterns? Not really. Actually, if we look at these, if we compare our expected to our observed, or look at the actual calculated differences between them, we see that it actually almost perfectly matches what we would expect to see. So I would say no. Data almost perfectly matches expectations. Lastly, for question 4, you are studying spiders living on different tiny Caribbean islands. You are interested in their leg lengths by different islands and want to see if there is a difference between the populations. You classify the leg lengths into 3 categories: short, medium, and long. Are the leg lengths similar between the 2 populations? And this is data in a CSV, so we need to import our data. I've already got it in my files area, so I'm just going to use file.choose. It's the chapter 14, question 4 CSV. Copy that, Control C, click in here, Control V, Control Enter to run that, and then head I'm going to check that data. Chapter 14, question 4. Got counts, populations, either population 1 or population 2, and our leg length, short, medium, or long. What test are we going to run? This is another test of homogeneity. And that's because we are actually looking at and that's because we are looking at 2 populations and 1 trait. So that was one of the things about homogeneity. 1 trait, 2 populations. Independence is 2 traits in 1 population. So our null hypothesis is observed frequency equals expected frequency. Observed frequency does not equal our expected frequency for the alternative. Specifically, we expect that leg length is similar between both islands. Leg length is not similar between both islands. So we've got our data in as an object, but now we need to make a matrix and do the test. So Q4 matrix, oops, not a space, matrix X tabs. We're organizing count data by population and leg length. The data we're using is the chapter 14 Q4. I'm then going to do the chi-square test. So Q4.chi, making an object out of it, chi-squared test, and I'm going to put the matrix inside of it. I haven't run either of these lines yet, so the matrix doesn't actually exist. And R tells me that with this yellow box. I'm then going to want to see the results of the chi-squared test and compare them. Chi-squared observed, chi-squared expected, and look at the difference between the two. Chi-squared observed minus expected. So again, I haven't run any of these lines yet, but I'm going to run them all together. And I just need to make sure that they're in the right order. That's what's important. Control, Shift, Enter. So we get our chi-squared calc, degrees of freedom, and our p-value, which is very small. So we're going to reject the null. P is less than 0.05. And again, this is the exact same thing as if I had run each of these individually. But by running them all together, I can keep seeing everything together. I don't have to run the lines before it again if I want to look at things again. Biologically, this means leg length differs between islands. Any patterns? So we are rejecting the null hypothesis. That means that our data doesn't fit the distribution we expected or isn't homogeneous in this case. So what is driving that? What are the patterns? Why are we seeing this difference? And we're looking for big numbers here. So right here, it looks like there's a little bit of deviation from short. For population 1, there's a couple more that have short legs than should. For population 2, there's a couple that have there's a couple of fewer that have short legs than they should. but the biggest deviation is between long and medium. It looks like population 1 has too many spiders with long legs and too few spiders with medium legs. Pop 1 has the opposite, or Pop 2 has the opposite, has too few spiders with long legs and too many spiders with medium legs. So essentially we could really sum that up and say Pop 1 has spiders with long legs compared to Pop 2, which has shorter legs. So this would be good. This would also be good.